despite the fact that we have the same DNA in all our cells, or having the same genome or set of genes in all our cells, but not all genes are expressed in all cells. Undoubtedly, there are some genes which are expressed in almost all our cells. We refer to those genes as housekeeping genes. Those are called housekeeping genes. But when it comes to other genes, different cell types, uh, different types of genes are active. For example, in mammalian cells, or the cells which actually give rise to RBCs, red blood cells, the gene for hemoglobin is active. The gene for hemoglobin is active. But what about other cell types? Is this gene active? No. Your muscle cells don't form hemoglobin. Your neurons don't form hemoglobin. So this gene is not active there. Same is the case. The gene that forms insulin in the beta cells of your islets of Langerhans in your pancreas is not active in other cell types. So different cells, when they differentiate during your what we say, when your different germ layers give rise to different cell types. So when the differentiation takes place, different cell types are actually differentiated on the type of genes which are active, and the type of genes which are not active, which are inactive. So one cell type differentiates from the other cell type. So in one cell type there are some genes which are active, but they might be inactive in other cell type. But the question is, the bigger question is, how is the expression of these genes regulated? What is the mechanism? How is it regulated? Now, before we get to that, let's understand at what levels the regulation of these genes or the regulation of the expression of these genes can be regulated. If you see, how do genes express themselves? Genes either express themselves to form proteins, so genes encode proteins in your cells, or in some other cases, genes do encode some non-protein molecules as well. Like genes can encode transfer RNA, tRNA, okay? Genes can encode ribosomal RNA, rRNA. Genes can encode small nuclear RNA, okay? So these could be the products of genes in your cells. So now the regulation of gene expression, it can be regulated at different levels. Now what are the levels where we can regulate it? When it, for example, in prokaryotes, if we reflect on, the genes are actually regulated mostly at transcriptional level. So this is your gene, or it is a segment of DNA. It encodes a messenger RNA. It forms a messenger RNA, and later on messenger RNA goes to the ribosomes, which actually are the protein factories, and messenger RNA is translated into a protein. Now, so this is from DNA, okay? It forms messenger RNA. This is what we call transcription. So this is transcription. So genes can be regulated at transcriptional level as well. Okay, this is what actually mostly happens in prokaryotes. They are regulated at transcriptional level. But when it comes to eukaryotes, genes can be regulated at, for example, in eukaryotes, uh, you might be knowing that post transcription, there is a modification that takes place, and one of the key steps in that modification is splicing. So regulation can happen at a splicing level as well. So regulation can happen at, so from messenger RNA it goes to the ribosomes, and protein is the end product. Okay, so this is what we call translation. So genes can be regulated at translational level as well. Okay, so they can be regulated at translational level as well. So they can be regulated, there is post translation regulation as well. So post translation. So genes can be regulated at different levels. They can be regulated at transcriptional level, they can be regulated at splicing level, they can be regulated uh, during translational level as well, they can be regulated post translational level as well. So there are different levels where the regulation of genes, the regulation of gene expression can be regulated. It can be controlled, it can be managed. Now, when it comes to prokaryotes, so if we reflect on prokaryotes, uh, there is essentially uh, a transcriptional unit which actually has got a cluster of genes which are expressed together, and they have the common a sequence of DNA which we call promoter. So they are actually they have the common promoter. Now see, we refer to this as operon. In case of prokaryotes, we refer to this as operon. Now see, let's get to that and check out how this operon looks like and what could be the possible genes that we have in the operon. Let me get here. If you check out, this is a bacterium, for example, a bacteria which resides in your gut, E. coli, for example. This is an E. coli bacteria, okay, which is a permanent resident of your gut. Now see, this bacterium, this is its genome, circular DNA. Now this is a sequence of DNA that we have taken. Okay. Now see, if you reflect on this sequence of DNA, this is the double-stranded prokaryotic DNA. This is the double-stranded prokaryotic DNA. This is a small sequence that we have taken. Now see, there are three genes here: gene 1, gene 2. Okay, so from here up till here, these are three genes: gene 1, gene 2, and gene 3. These are essentially structural genes. And these are structural genes. Okay, so these genes encode proteins. So protein 1, protein 2, and protein 3. These genes encode proteins, and these proteins have a function other than regulation. Okay, they have no role in regulating, okay, this operon. So that's why we use the term structural gene. Okay, so structure, when we say structural gene, so structural genes actually encode proteins. They encode proteins, and these proteins have specific function. Okay, usually these proteins, which, because these genes are transcribed together and later on translated, so these proteins, they are involved in the same function. For example, different steps in a metabolic pathway. Okay, so they are involved. Now see, along with these genes, there are some DNA sequences here, and these DNA sequences are referred as regulatory DNA sequences. These are regulatory DNA sequences. Now, what we understand by these regulatory DNA sequences? Now, see, these regulatory DNA sequences actually refer to those sequences which actually regulate the transcription of these structural genes. Now, see, there is this sequence here. Let me show it with blue. So there is this sequence here. This is essentially one of the regulatory DNA sequences, which we call promoter. So promoter is essentially a regulatory DNA sequence, and this is the site where the RNA polymerase, which actually is responsible for transcribing these structural genes, this is the enzyme that is responsible, that binds to this site. Okay, so this is the promoter site. So again, so promoter is the region where regulatory DNA sequences is a regulatory DNA sequence where your RNA polymerase binds, and later on it brings about the transcription of these regulatory genes. Okay, now there could be other sequences as well. For example, there are certain molecules, small molecules, which actually bind to these regulatory DNA sequences, and they either stop, or they either promote the expression of these structural genes. And if they promote, we call them inducers, or you can call them activators. Okay, so this site over here, let me show it in green. This is the site that we call operator site. This is the operator site. Okay, so this is the site where a molecule can bind to this site. Okay, and it can block this RNA polymerase, and it can stop the expression of these structural genes. Okay, so usually the molecule that binds to this site and stops expression of these genes, we refer to that molecule as a repressor. That molecule is referred as a repressor. That molecule is referred as repressor. Okay, but there can be other molecules as well which will facilitate RNA polymerase to bind to the promoter site and carry on transcription. For example, if this is the molecule here, okay, it's a small molecule that binds to this regulatory DNA sequence here, and it facilitates the binding of RNA polymerase to the promoter site, and later on brings about the transcription of this operon of these genes. Okay, so this will be referred as activator. This will be referred as activator. Okay, now operon is essentially a cluster of these structural genes with these accessory regulatory DNA sequences. Okay, now there could be other DNA sequences as well. Okay, which actually form certain proteins which actually repress or stop this RNA polymerase from transcribing these genes. Okay, that is not we refer to those regulatory sequences. Okay, those sequences, those regulatory proteins, those are regulatory proteins. So that's not a part of your operon. Okay, so that is not a part of operon. Now when it comes to these operons, okay, in these bacterial cells or prokaryotic cells, these operons can be let me get to the other page. These operons this these operons can be when you have these operons can be inducible, or these operons can be repressible operons. Now, what is the difference between inducible and repressible operons? Actually, inducible operons are those operons which by default these operons are off. Okay, then a small molecule comes. Okay, in presence of a small molecule, this operon is turned on. It turns on. Okay, so in presence of a small molecule, like we will study later on in case of lac operon, okay, in presence of lactose, the operon turns on. So this uh, operon, okay, this small molecule, in presence of which this operon turns on and the structural genes are transcribed, okay, so this is called inducer. This molecule is called inducer. Okay, so we refer to this operon as inducible operon. Now see what you understand by repressible operon. So repressible operons by default are on. So by default these operons are on. Okay, the genes in these operons, the structural genes, they are transcribed. Okay, so I would like to repeat over here that in an operon the structural genes are transcribed together. Okay, and they have the same promoter, they have the common promoter. Now see, these repressible operons, they are usually on, they are by default they are on, but a molecule in presence of some small molecule, if they are turned off, if this operon turns off, okay, in presence of some small molecule, so that stops this operon from transcribing the structural genes. Okay, so this will be referred as a repressible operon. We refer to that operon as repressible. So by default inducible operons are off, okay, and a molecule comes and turns it on. Repressible operons, by default the operon is on, a small molecule comes, okay, which we later on will discuss in case of TRP operon, which is repressible operon. So the operon is turned off in presence of a certain molecule, a small molecule. So in this is for today an introduction to regulation of gene expression. Let's revise the key points. Okay, in prokaryotes, the gene regulation mostly happens at transcriptional level, but in eukaryotes, if we reflect on eukaryotes, this regulation can take place at different levels. It can take place at transcriptional level, splicing, which is a post-transcriptional phase. It can take place at translation level or post-transcriptional level, uh, and then in prokaryotes, actually there is a cluster of genes along with some accessory DNA regulatory sequences which operate together, and we refer to them as operons. When it comes to these operons, there are structural genes and regulatory sequences. So these operons can be inducible or repressible depending on whether they by default are off or on. And there are some molecules if it is by default off, so when there is a need, so the cell will turn it on, and this will be turned on due to the presence of some small molecule, and when the cell needs to turn off a particular operon, so a small molecule will come in and it will turn the operon off. So such operons are referred as repressible operons. Thank you.